We're just going to go ahead and give the attendees some time to join, and then we'll go ahead and get started. All right, everybody, we're going to go ahead and get started with tonight's session. My name is Michaela Clark, and I am one of the co-chairs for the Research Symposium this year. We are really excited to hear about our keynote speaker, as well as announce awards for all of our presenters. We would also like to take this time to thank Zions Bank and Practice Pathways for sponsoring this portion of the symposium. Practice Pathways is a special initiative with Zions Bank to assist healthcare providers in establishing their own medical and clinical practices. We have joining us tonight, David Kirby, who is the Senior Vice President for Zions Bank and the Director of Practice Pathways. We also have Ray Swager, who's the Senior Vice President for Zions Bank. We would like to thank them for sponsoring this part of the symposium.
Thank you, Dr. Clark. We would now like to introduce our keynote speaker. After his presentation, we will be taking questions from the audience. As you might remember how we do with our podium speakers, we will be taking those questions at the end of his talk. And just remember to add them on the Q&A chat box that is located on the bottom of your screen. Our keynote speaker tonight is Dr. Larry Schlesinger. He's internationally recognized authority in infectious diseases and was recognized recently in San Antonio, Texas, by the World Affairs Council as the International Citizen of the Year. His passion lies in the development of people and programs that will have revolutionary impact on human health for years to come. He began his distinguished scientific leadership career after joining the faculty at the University of Iowa in 1991. Later, he moved to Ohio State University in 2002, where he served as director of the Division of Infectious Diseases, Department of Internal Medicine until 2011, where he became first chair of microbial infection and immunity. He was also founding director of what is now the University-wide Infectious Diseases Institute. In 2017, he became professor, president, and CEO of Texas Biomedical Research Institute in San Antonio, Texas. His discoveries have led to greater insights into the unique attributes that soluble and cellular components of the innate immune system on humans bring to the microbe-host interface, translating them into drugs discovery platforms. Now we're really excited, Dr. Larry Schlesinger, to hear from your talk. The time is yours.
Okay. Sorry, I'm trying to put it in slideshow. Okay, do we have it now? Can you
Yes,
see
it
it?
looks right.
And can you hear me?
Perfect.
Okay, great, great. Well, good late afternoon, evening, everyone. Thank you all for coming. And I'm so pleased to have the opportunity to speak with all of you this evening. It's a tremendous honor. And there is an individual, a postdoc in my lab, who's very familiar with Roseman and speaks so highly about your organization. So I'm really pleased to have the opportunity to talk to you about the impact of past, present, and future pandemics, something that I'm very involved with and very interested in, and I hope you'll enjoy my presentation this evening. What I'd like to do in our time together is to give you a very brief overview of pandemics, because I think we need to set the stage. I then want to spend most of my time this evening speaking with you about COVID-19, the science in particular of yesterday, today, and tomorrow. I then want to spend a very brief amount of time talking about the intersect, and I will say impact, of COVID-19 and other infectious diseases. And then I'm going to end by telling you how a small Texas nonprofit research institute, that is Texas Biomedical Research Institute, is taking on the challenge of eradicating infectious diseases in the world. So here I want to start with a little history in pandemics. Infectious diseases have arguably been the single most important factor in human evolution. Historically, major pandemics have killed tens of millions of humans. Now, microbes are the earliest life forms. So they've learned to adapt well beyond the adaptations of humans and are able to do so very quickly. Therefore, epidemics and pandemics have plagued humankind since the beginning. Settlers in the 19th century often spoke of cholera, typhoid, yellow fever, and other illnesses, causing towns, especially port cities, to quarantine. But some of the earliest epidemics actually date back thousands of years, and we don't even know what the cause was. So in 3000 BC, an unknown epidemic ravaged a civilization in China that wiped out the entire population, leaving archaeologists to determine that the epidemic was so quick that the dead were not even buried properly. The earliest recorded pandemic was in 430 BC in Athens, Greece, and was a major player in the defeat of the Spartans. And to this day, we don't know whether it was measles, Ebola, or typhoid, perhaps. But throughout history, disease outbreaks have ravaged humanity, sometimes changing the course of history, and at times signaling the end of entire civilizations. And with all of that, now we have the pandemic of COVID-19. I want to compare today's COVID-19 pandemic historically with the pandemic of influenza of 1918. So the 1918 influenza pandemic was the most severe pandemic in recent history. It was caused by a strain of virus called H1N1 and had genes of avian origin, so otherwise known as avian influenza. Although there's not universal consensus regarding where the virus originated, it spread worldwide over 2 years, 1918 to 1919. In the United States, it was first identified in military personnel in the Midwest in the spring of 1918. It's estimated that about 5 million people, or about 1/3 of the world's population at the time of 1.8 billion, became infected with this virus. The number of deaths was estimated to be about 50 million worldwide, and about 675,000 deaths occurred in the US, and we compare that now with over 560,000 deaths due to COVID-19. Now, interestingly, in 1918, the mortality was very high in young individuals below age 5. It was also very high in individuals 20 to 40 years old and in the elderly over 65. The high mortality in the healthy individuals was unique. Not the same as now. Can you all still hear me?
Yes.
Okay, great, thank you. Because I got a little note that it had changed. So that's a unique feature of the 1918 pandemic. Now, in 1918, with no vaccine to protect against influenza infection and no antibiotics to treat secondary bacterial infections that were found to be associated with the influenza pandemic. Control efforts were limited to non-pharmacological interventions, such as isolation, quarantine, good personal hygiene, use of disinfectants, and limitations of public gatherings. And guess what? They were applied unevenly. Does any of this ring a bell with today's pandemic? There was not much science going on in the United States in 2018. And if anyone ever picked up the John Berry book on the Great Influenza, you'll learn that it was the pandemic of 1918 that really launched American science. In fact, the story of American science in the 20th century is just an extraordinary story, the development of Johns Hopkins University, the Rockefeller Foundation, and others. And so there was the rapid development of the beginning of interventions. That is another distinguishing feature of the pandemic, and one that I'll come back to now with regard to COVID-19. Now, when looking at the number of deaths in the US, it helps a community to come to terms with the severity and magnitude of any historic event. And what I'm showing you here on this slide is that looking at US deaths compared to war deaths, COVID-19 has now surpassed the largest war-related deaths by more than 50,000. Again, I'm just checking to make sure that you can see the slide. Hopefully you can.
Yes, Dr.
Yes.
Schlesinger, we can see your slide, and your video cut out for a second, but we can see you now. Oh, it cut out again.
Okay, hold on.
There you go. We see you now.
Okay.
Perfect.
Okay. All right. Okay, great. So with that as a very quick backdrop, I want to now segue to COVID-19. If I can get my slide to transition. Sorry about that. Okay, now I think we're back on track. So the wonders of a virtual conference, sorry about that. I'd like to take a quick step back now and remind us all of where we began with regard to the COVID-19 pandemic. So I want to show you this timeline and have you recall that it was January 3rd in Wuhan, China, that there was notification to the World Health Organization of an outbreak of a virus that right away was known as a coronavirus. We know now today that in the fall of 2019, that there was already outbreaks or pockets of individuals getting sick, likely with COVID-19 before that time. The first American death was on February 6th, and the World Health Organization declared a pandemic on March 11th. Borders began to close in March, and by July, vaccine trials had begun extraordinarily fast. And I will tell you as a forecast that Texas Biomed was very active in that period of time. I'll come back to that. So vaccine trials in July, vaccines approved in December. At the time that vaccines are approved, we're learning about variants, COVID-19 variants. And if we look at where we are today in the upper right, we know that cases have declined. I'll show you that in just a moment, but they're still high. The vaccine rollout is getting better, although there are fits and starts, and we have a battle with increasing variants that are around. So cases have declined, but we're still at about 60 to 70,000 cases a day, which is way too high. And according to the CDC, as of April 9th, the United States is in the fourth week of an upward trend in COVID-19 cases. Now, the lifting of physical distancing and mask mandates in some areas could certainly be contributing to what we're seeing today, that's for sure. And the increase can also be due to the presence of the more contagious variants of SARS-CoV-2. And on the left, I'm showing you a variety of variants that have been identified. We know that B.1.1.7, initially identified in Great Britain, is the most common variant circulating in the United States today. and has been reported now in all 50 states, including the District of Columbia and Puerto Rico. And we know that the vaccines are rolling out at an accelerated pace, and there is now a race between vaccination and the emergence of these highly infectious variants, something that we need to pay attention to. So as we like to say, there's a light at the end of the tunnel, but we're still in the tunnel. We need to be very careful, and that's very important today. Now, I want to transition to say, where is science now? I think you all know that there is a research pipeline for vaccines and therapies, but I'll show you some things that perhaps you don't know. We have clinical vaccine and therapy trials. I'll show you that in just a moment. And then I wanted to segue a little bit to preclinical research and the importance of preclinical research in getting us to a shot in your arm and where we are today. So on the right is a so-called coronavirus vaccine tracker. And what I wanted you to know is there are currently, and you may not know this, 86 vaccines in clinical trials. And that there are 77 vaccines that are in the preclinical space under research development to potentially be a vaccine for the future. And I will tell you that this very active preclinical pipeline will be essential for us as we think about vaccines, next generation vaccines of the future. You might not know that there are over 590 drugs in development or in planning stages in the preclinical space. We have a small set of approved clinical drugs, mostly the monoclonal antibody therapies that are available today. But if you look at the upper right, you'll see that now there's being developed more specific antivirals, cell and gene therapies, so-called immunomodulatory therapies, and other types of therapies that are rapidly becoming on the scene. And I will tell you that in addition to vaccines, the therapies are absolutely critical, because even as we speak today, there are sick patients in the hospital who need more effective therapies. The monoclonal antibodies are effective to some degree, but they're limited in their utility. They're intravenous and they have to be given at a very specific time. So we need more therapies, and that's very important going forward. But I want to focus a little bit now on the preclinical space, because this is something that general audiences think less about. So what is the preclinical research space? It's scientific disciplines that traditionally have been studied before introducing patient care. And the aims are to understand basic biological processes, host pathogen interactions, and to test the safety and efficacy of diagnostics, therapies, and vaccines. Here is a slide of a typical drug discovery pipeline that I wanted to show you. And the bottom line is that the typical drug discovery pipeline is at a minimum 12 to 15 years, and it's very expensive. It starts in basic science laboratories with identification of targets for any new therapy or vaccine. It then moves to the development of in vitro assays for target assay construction and screening. And then one ultimately has to get to lead compounds that are potentially therapeutics. One needs to bring in medicinal chemistry, and one needs to think about validation and safety. And this takes quite a bit of time, and sometimes is slowed for a number of reasons, as I'll show you in just a moment. We then get into the clinical phases, of which there are actually four. The fourth phase is research. Okay, is for post-marketing research. Phase 1 trials are typically very small. They're 50 to 100 people, and they're really focused on safety. Phase 2 are just hundreds of people, and it's mostly safety with some efficacy. Phase 3, as you've been hearing a lot about, are tens of thousands of people. And this is really where one proves whether a new therapy or vaccine is safe. And then all of this material, not just the clinical phase trials, but the preclinical information gets submitted to the FDA to get even emergency use authorization. And what the public doesn't really understand often is that the FDA will come back and ask for more science. So it's a back and forth iterative process that can go through several phases. And I will tell you that we did all the preclinical work on the Pfizer vaccine. We're very proud of that, but we did 7 studies till the FDA was comfortable. This is a lot of work back and forth to get something, a shot in your arm. Okay, I think when I lose it, I have to get out and come back in. I'm sorry about that. So what I'm showing you, I showed you the typical pipeline of 12 to 15 years. And now what I'm showing you is specifically the use of animal models in the preclinical space. So let me say that animal models are absolutely essential for any new drugs and therapies that come on the market. We are developing new models, organs on a chip, and ways to actually supplement or complement animal studies. But as we speak today, and as evidenced by COVID-19, they're absolutely essential. So following this pipeline, one starts with really small animal research, doing proof of principle type research with compounds that can potentially become therapies or vaccines. Then one moves on to more sophisticated animal models. And what's critical here is that the animal models recapitulate human disease. And then ultimately one has the validation studies in those animals to be able to move forward with clinical trials. So the importance of animal research is obviously in testing new drugs and vaccines, but animals are the absolute way to discover how diseases occur. They help you understand transmission of pathogens, virus or otherwise, the risk of reinfection, and a so-called antibody-mediated immune enhancement, which is really something that can occur when you do generate a therapy or a vaccine that involves antibody production, and it's an adverse effect that you'd want to know about in animals early. There are a lot of considerations that go into animal research: the proper animal model that recapitulates human disease, the cost, the number, the challenge strain, which virus you use or viruses you use, the dose and the route of administration, how often you give it, age and sex, of course, the ability of the virus to mutate and change and how that impacts response. And the animal rule really applies to things like Ebola, which Texas Biomed also studies, where you really can't do human studies. So you have to do all your studies in animals. And of course, the need for biocontainment to do all this research. There are a number of readouts, clinical, virologic, immunologic, and histopathology readouts that are critical here when doing animal research. And then one needs to reflect on standardization or so-called GLP or good laboratory practice science. And this is critical. Perhaps you didn't know that the US spends nearly $60 billion in preclinical research each year. And up to 50% of the preclinical research is subject to reproducibility errors. This is something that's been written up a lot about in Science and Nature. So there's a real focus by the FDA on GLP level science, that level of rigor that allows them to be comfortable that the data are real. So this is an extremely important part of animal research and something Texas Biomed does a lot of. Now, having said everything to this point, I have a slide here called The Winding Road of Academic Drug Discovery, because it doesn't happen the way it's happening with COVID-19 usually. So what this model shows is the ability to move from discovery to phase 1 trial success. Now, if you look, you don't have to look at this very complicated slide, but if you just look at the blue ovals represent the pathway to phase 1 trial success. The green circles on this are kind of go, no go. So in any development of a drug or a vaccine, one has absolute block points where you think some drug is going to move forward or not, and that's pretty clear. But the key point of this slide is look at all the pink structures here. They're typically more pink than blue and green, and that's because typically, and this is often in academic research, there's derailment. Now, why is there derailment? So there can be money issues. There often are. That's the problem with moving translational science forward. A scientist leading a project can get a better job at another university, and the science gets interrupted. There's often slowdown in tech licensing, perhaps lack of availability or expertise in getting intellectual property solved. And then it's this issue of validation. So oftentimes this science either stops or slows. It's been called kind of the abyss in trying to develop effective drugs and vaccines. And that's really important in thinking about moving forward in science translationally. So what does the future look like? So it's something we began to experience, perhaps still in a reactive mode, and that is Operation Warp Speed. But this is the idea that academia, the private sector, the public sector, research institutes come together to develop a more streamlined process to success on diagnostics, therapies, and vaccines. So linking organizations, having them lean in and be working, as I'll say in a moment, proactively to enhance the R&D, the research and development pipeline, and have sustainable finances so that potential drugs can actually get to the finish line. And there are examples of several efforts in drug and vaccine development that never made it to the finish line. In fact, vaccine started being developed for SARS back in 2003, the first SARS outbreak, and that one, luckily the outbreak stopped, but the vaccines actually never got finished. Having said all that, and in thinking about my previous slides, on the top bar and the bottom is the normal 12 to 15-year process that's on target antigens, animal models, all through the phases to get to a shot in your arm. And with SARS-CoV-2, we experienced an extraordinary scenario. In fact, it's the hero of the day, in my opinion, that we were able to move so quickly to get to a vaccine. But I have to warn you that this was absolutely the best case scenario, and we will perhaps not be so lucky when the next pandemic hits us. So decades of research by tens of thousands of scientists worldwide actually put in place the essential knowledge and methods that underpin the rapid development of the COVID-19 vaccine. So it starts with the fact that we did have a SARS outbreak and we understood what mutation of coronavirus family means. And then we had an outbreak in 2012 with MERS. And so we knew the virus. In fact, and perhaps you don't know that about 30% of upper respiratory infections in humans each year is due to common coronavirus. We know a lot about them. And so because of that, there had already been a lot of work on spike proteins for other coronaviruses. So we knew how to design a SARS-CoV-2 version of that spike protein, which is the focus of vaccine development. So we knew a lot about the virus. And right from the beginning, out of Wuhan, China, we knew it was a coronavirus. As you know, that even with SARS, it began with people getting fever. We didn't know what the microbe was, and it took about 3 months to identify it as a virus, as a coronavirus. So even there, we were able to short circuit. We also were able to validate animal models very quickly. I'll show that to you in just a few moments. And then I want to talk about these vaccine platforms. So the mRNA vaccine platform has actually been around since the 1990s. And it was being developed in several companies for use for influenza, HIV, and other types of viruses. So it actually was available as a platform to pivot for COVID-19. And the adenovirus vector, you know, the basis of the Johnson & Johnson vaccine, work on adenoviruses began in the 1970s. So we know a lot about them. In fact, I was indirectly involved in some research in the 1980s at the University of Michigan that used adenovirus as a vector to deliver genes, in that case, to animals before humans. So we had a lot of background that enabled us to move so quickly. We had everything in place, and we were able to exploit our scientific knowledge so that we could have a shot in your arm quickly. Well, where are our challenges today with COVID-19, in my opinion? I think still the greatest challenge with this virus is still understanding how the virus causes disease. So we're actually developing interventions at a time we're still trying to figure out what the mutation of SARS-CoV-2, what it means in terms of the host response. So what we do know now is there are two phases of COVID-19 infection. One is the virus, that is the acute infection. And then the second part of COVID-19 is the inflammatory response that's gone awry in humans who develop COVID-19. And that's really important because it really talks about different ways of treatment. Now, related to that is the long hauler syndrome. That is up to 40% of people who have chronic symptoms after they've had COVID-19. In fact, I think this is one of the biggest motivating factors for getting a vaccine because of the risk of long-term complications. The triad I talk to everyone about when I speak is that one needs to think about infectious disease differently. Microbes trigger immunity and inflammation triggers a variety of diseases, be it cancer, heart disease, brain disease, autoimmune disease. So the triad of infectious agent as a trigger, inflammation as a host response, and then the development of chronic inflammatory disorders is something that everybody needs to know. And this is something we're still learning about today. I've already mentioned the variant strains that are a real challenge for us as we're trying to have the race against these variants and their infectivity. We're having, as would be predicted, some problems with vaccine rollout, and that's because we're in a crisis mode and we're reacting. We didn't have the manufacturing plants or the organization nearly where it needed to be before the pandemic hit. We have this issue of vaccine hesitancy, which I'm going to talk about just a little bit more in a few minutes. For me, as an infectious disease person who's been through several of these outbreaks, the politicizing of public health has been extraordinary. The rapid information exchange each day, the disinformation is new. It's putting a premium on education, and I speak all the time on this to bring facts to the table because of this misinformation, and that's been very, very difficult all along. And then finally, I want to talk about this reactive versus what I think the future needs to be, because there will be another pandemic. I don't know if it's coronavirus, but there will be, and we need to be proactive. So we need to change the way we think about infectious diseases. Now, the anti-vaccination movement is here. We learned about it before COVID-19. It was actually occurring in the setting of our measles, growth in measles outbreak in the United States. And it really began in the 1990s. There was a landmark article, if you can call it that, in 1998, on 12 cases of autism that were suspected but not proven to be linked to the MMR vaccine, and subsequently completely shown to be false. But really, that began this process of mistrust. There's also this mistrust of the pharmaceutical companies. They're just in it for the money. And also then there's the belief that perhaps, these infections are not serious. I don't need a vaccine. And there are several other reasons as well. It's important to point out that this anti-vaccination movement or vaccine hesitancy is a global, not just a US issue. And it is deemed to be a top 10 international health problem by the World Health Organization. So this is really a big problem for us going forward. And what I need to say is that vaccines historically have prevented hundreds of millions of deaths due to infectious diseases related to polio, measles, hepatitis, meningitis, saving up to estimated 3 million lives a year. So vaccines really have been monumental in terms of us protecting the public from infectious diseases. Now, I mentioned this reactive versus proactive. This is a peer-reviewed journal, Time Magazine, not really, from 2017, but it's a good article. And the bottom line of the article is that we're not prepared for these pandemics. And it's interesting because I presented this picture to the Board of Trustees when I took the position at Texas Biomed in 2017, saying there will be a pandemic. So now you can imagine the board sees me as somewhat prescient in terms of thinking about infectious diseases. Now, all of us feel that we agree that while it's become possible to control or eradicate certain infectious diseases, it seems unlikely we'll be able to eliminate most emerging infectious diseases in the foreseeable future. One economist from Britain has predicted that infectious disease is the #1 killer of humans by 2050, more cases than cardiovascular disease, to the tune of an extra 10 million lives a year, or an additional $100 trillion to the global debt each year. This is really a big growing problem that we need to pay more and more attention to. This is an article that was published in the Proceedings of the National Academy of Sciences that I use, by David Bloom. And I think it's a very important article. And what it does is it identifies the normal response to an infectious disease outbreak. So in the top right, if you can see my pointer, it starts with some cases of people getting sick with fever. There's then some articles written about those cases from media. Money starts pouring in, and then we start getting a lot of people leading in to develop interventions. But the key here is that we reacted. We weren't able to get in front of the first cases. And when the project ends and when COVID-19 goes away, and it will, then public attention goes away and money dries up. The future really does need to be thinking about infrastructure change that allows us to have 24/7 operations in the public and private sector that allow us to be better prepared for pandemics. And this is really being discussed at every level. If you've listened to Bill Gates recently, he talks about this, as well as Tony Fauci and others. So I do think we do need to think about a proactive response going forward. Now, the next point I wanted to make was that there was an article published in the New York Times, actually an excellent article just within the year. It said the biggest monster is actually not COVID-19. It's the other scourges of our time: tuberculosis, malaria, HIV, and other infectious diseases. And I'm going to briefly take you through those right now. But the bottom line is there's not enough time, money, or resources to handle all of these major scourges. And in the setting of COVID-19 pandemic, a lot of the infrastructure to handle, for example, tuberculosis has been shifted over to COVID-19. So we can really look forward to an aftermath, because there's a lot of cases of these other infections that aren't being diagnosed today as a result of our focus on COVID-19. So in the case of HIV, this is the virus that causes AIDS. More than 37 million people have HIV around the world. It's a global epidemic that continues to claim lives today, about 1 million every year. Now, while effective treatments for HIV exist, there's no vaccine and no effective curative treatments, so that we've come a long way in helping people live healthier lives with HIV. But still, many people in the world don't even know about their HIV status and continue to spread it to others. So this continues to be a major problem. The next infection I wanted to just briefly mention is malaria. Malaria is a parasitic disease transmitted to humans through the bite of a mosquito. It also remains a major killer worldwide, particularly of children. And there's a growing problem of parasite resistance to anti-malarial medicines and mosquito resistance to insecticides. And again, unmet financial needs. And so there had been a lot of progress in the late 2000s and early 2010. But really, innovations, prevention, treatment have largely stalled over the past few years. Today, more than 90% of the world malaria deaths occur in sub-Saharan Africa, and most occur in children under age 5. Where only 1/3 of the children with fever are even taken to a medical provider. So clearly, the pace of progress for malaria needs to increase. The disease I study is tuberculosis, and it is, again, continues to be a major scourge of our time. To date, 1.4 billion people have died of tuberculosis, greater than any other single infectious disease threat by a long shot. 1.5 million people die of tuberculosis every year. It's caused by human transmission of a bacterial pathogen. Of the 10 million people who become ill with TB each year, 4 million don't get the care they need. And there is a huge problem with drug resistance. So my own research is in figuring out how TB exploits our immune system in the lung. And a lot of my research translates into what is called host-directed therapy, which is a new approach that handles this inflammation issue simultaneously with drugs, that is antibiotics, to treat tuberculosis. And again, there's a major gap in funding. And finally, I think we all know about Ebola. We've had increasing outbreaks of Ebola in the world. In Western Africa, the largest outbreak ever was in 2014 to 16. Then beginning in 2018 in the Democratic Republic of Congo, there was an outbreak that kind of slowed down. And then there was more outbreak in the DRC. And now we're just beginning to hear about more cases in Guinea back to Western Africa. Now, I talk about Ebola because Texas Biomed has spent the last decade or more on therapy and vaccines for Ebola, and we actually played a major role with our preclinical studies in the new drugs and vaccines that are being used in Africa today. So let me close by saying, what is a small place like Texas Biomed doing in this arena? I've alluded to us a few times now. So we're a freestanding nonprofit research institute in a beautiful section of San Antonio. Think about that when you think about an infectious disease research institute. We have 370 employees. We do have close working relationships with the medical school in town, as well as with the undergraduate campus, UTSA. So we are the only private nonprofit research institute in the US to combine the following. We have 7 high biocontainment facilities on our 200+ acre campus. Two of these are CDC regulated labs, and the only privately owned biocontainment level 4 lab in the country. The rest are federal labs. There's about 10 total, where we do all of our hemorrhagic virus Ebola type research. We have one of 7 national primate research centers, the only one that's not on a university campus. We have 150 employees dedicated to the humane use of animals on our campus. We have decades of experience in what is called regulated GLP-like science that I've talked about, and the FDA knows us well in terms of the quality of science we produce. And we have a very entrepreneurial culture that really allows us to think from bench to bedside in a real way. So the bedrock of our organization is fundamental research, NIH type research, but at the same time, we have a contract organization where we can really bring diagnostics, therapies, and vaccines to market. And we have a history of success with the COVID-19 vaccine. I'll give you an example. We were involved early on in Ebola treatment. We were the company that studied Gilead's cure therapy for hepatitis C. The Nobel Prize was recently awarded to 3 scientists who discovered hep C. All of them did their chimpanzee work, their animal work at Texas Biomed. And we've had a number of other discoveries over time. So it's a very unique place. Let me give you an example of how we can work and how quickly we can work. So we started learning about the COVID-19 epidemic, and we started discussing this and brought a team together because we don't have departments, we have research programs. We brought an eclectic team of researchers together very quickly in late February, early March, and said, we want to make a contribution. Where can we do that? And we knew that we could do it by developing some of the earliest validated animal models for COVID-19 because we're good at that. We went out and had a campaign, and within a week or so, we were able to obtain $5 million from contributors and launch our small and large animal studies. We ended up with 4, 2 models in primates and 2 in small rodent models. By May, we were writing papers on these models, which is the bedrock of fundamental research. And between May and June, we entered into partnerships with Pfizer for their vaccine, Regeneron for their monoclonal antibody cocktail, Novavax, which you'll be hearing more about. It's a vaccine that'll come on the market shortly, I believe. And then by August, the vaccine studies and therapeutic studies were all complete, more papers. We had approval of the Regeneron monoclonal antibody therapy and the Pfizer vaccine in December. And now we have over 35 contracts, large business going on with related to approximately 30 companies doing work on next generation therapies and vaccines. So this is really how it works. And we were working in parallel with these companies as they were pushing forward on their clinical trials to accelerate the movement towards a FDA-approved vaccine or therapy. So this is just some of the work that we were involved in. You hear about the former president receiving the Regeneron monoclonal cocktail and our work with Pfizer on their vaccines. The Regeneron monoclonal antibodies we published with the company in Science magazine, so it's very important we're able to produce this work in very high impact journals. So we really are positioning ourselves as global leaders in infectious diseases and taking on global challenges. And we do that because we have tremendous strength in diseases that really greatly influence humans in the world. In terms of malaria, we're characterizing drug resistance using model systems and genetic engineering, pinpointing transmission reservoirs in the world, in Africa, in Asia, and developing new interventions based on that knowledge. In HIV/AIDS, we're very involved in vaccine development and testing, finding new ways to deliver therapies, so-called mucosal therapies, and now studying the use of cannabinoids to subvert the inflammation part of what infectious disease causes. In tuberculosis space, we have nearly 40 investigators now working on causes of disease and transmission, new diagnostics, therapies, and vaccines, preclinical and field studies in diagnostics in particular. In Ebola, as I've mentioned, we've been involved in Ebola research for nearly 20 years, involved in characterizing models of these diseases, testing therapies and vaccines. Our virologists are using reverse genetic engineering to develop new generation vaccines with an eye towards universal vaccines for the entire coronavirus family as well as for influenza. And then finally, really what infectious diseases is, it's really targeting those who are vulnerable. The Institute is as much interested in vulnerable populations that have an abnormal inflammatory response to a virus as it is the virus itself. So we have studies going on in the elderly, diabetes, those with immunocompromised systems such as cancer. So think about COVID-19, these are the target populations where there is a lot of severe disease going on. So let me finish my comments today by saying that by providing collaborative state-of-the-art science that delivers quality results in an efficient, time-efficient manner, Texas Biomed is making life-saving discoveries possible, I believe, in a timely fashion. And this is exemplified by our work on a variety of infectious diseases that I've already mentioned. And as I've said, we're unusual in that we're a hybrid organization. We're executing basic research as well as supporting the contract arm of our organization. And on both sides of the equation, our focus is on delivering results in a non-bureaucratic, time-efficient manner. And by being the CEO as a scientist, really that is my focus. And I'll end there and happy to answer questions, practical questions on COVID-19 or anything I've spoken about today. And there is my email address. If we don't get to all the questions, please write it down and feel free to reach out to me. I'm happy to address any questions you might have that we don't get to now. Thank you very much for your time. I'll turn it over to you now.
Thank you so much, Dr. Schlesinger, for your wonderful presentation on pandemics and telling us a little bit more about Texas Biomedical Institute. So we would like to take questions from the audience now. If you want to please use the Q&A box that you can see at the bottom of your screen. All right, so we have our first question here, Dr. Schlesinger. So having had both cancer and COVID-19, the vaccine trials you are currently running, do you feel there is potential risks for developing additional immunocomplications post-vaccination in the cancer population in the long term?
You know, so I mean, long term is a relative, a relative time scale. None of us in infectious disease research or otherwise can speak about longer term because we're just in the process right now. But in the phase 3 clinical trials, individuals with immunocompromised conditions were involved. We've now, as you know, vaccinated tens of millions of people. There is no indication that those individuals who have had cancer have an unusual response to the vaccine relative to the regular population. Now, having said that, anyone with cancer needs to speak with their primary physician about whether the risks outweigh the benefits of the vaccine. But that discussion must be had. And to date, the lead vaccines, Moderna and Pfizer, appear to be functioning very safely in a very large population as the vaccines are rolling out. So, you know, I can never really have a crystal ball about what long-term consequences might be, but I will tell you this. If someone with cancer has COVID-19 and is a long hauler, I think the outcome, might be more serious than having the vaccine. So that's how I would address that, Michaela.
The next question that we have is, I'm interested in learning more about animal testing practices. I understand its importance to the process of making safe medications for humans. Can you describe the humane animal testing practices that are used at your facility? Thank you. Sure,
I'm happy to do that. And let me start by saying that this is by definition a sensitive topic, and I'm very aware of that. And in fact, when you read about Pfizer and Regeneron, you'll really, in the newspaper, Wall Street Journal, New York Times, you'll read about the clinical trials and not about the preclinical research that allowed for those clinical trials to occur. So a National Primate Center is a major organization which is highly regulated by the National Institutes of Health and other regulatory agencies like the CDC. We have regulatory agencies on our campus, sometimes almost monthly, to ensure that we are providing the most humane care of our animals. We have a team of behavioralists that are specifically on campus to ensure that our animals are safe, healthy, and receive excellent care. So this is a big part of the way we do our research in taking care of these animals. They're precious, and we spend a lot of time making sure that those animals are safe. We're very proactive in describing that to the public because we believe it's important for the public to understand that, because sometimes there's misunderstanding and communication based on some unfortunate publications, et cetera, about the use of animals. So that's what I would say. And those studies are scrutinized that we do in animals. We make sure we use the minimum number of animals necessary, and that our studies are done in a very standardized way, so everything counts. So those are the important elements.
We have another question. What are we doing as a country to better prepare for pandemics?
Yeah, I'm hopeful, but I don't know yet whether COVID-19 will really fundamentally change the national landscape of preparation for infectious disease outbreaks. Pandemics are the focus of the day, but the number one killer in our hospitals today in the intensive care units, for example, is sepsis, bacterial sepsis, and that hasn't changed in many decades. Antimicrobial drug resistance is growing. We're losing drugs to infections that treat our neighbors in the hospital. There needs to be a sea change, and it's really going to be about financial sustainability and new organizational structures that allow us to be working proactively. One of the ways that I think we may transform this arena is that understanding how pandemics in real time now shut down society, that we've raised pandemics to the level of national security. If we think about national security and the way we finance national security and protect the US and the world from national security threats, maybe this is the way we need to start thinking about infectious disease threats. In the state of Texas, I am involved with a couple of senators in promoting a bill for an organizational structure in Texas that will allow us to put a large amount of money into proactive diagnostics, therapies, and vaccines, and public health for infectious diseases. So I'm hopeful, but time will tell.
We have a question from Dr. Gillis in the audience. Dr. Gillis, if you'd like to unmute yourself.
Hi, thank you very much for a wonderful presentation, but I actually don't have a question for you. I was very pleased, though, that we did have an ethics question asked.
Absolutely.
I apologize, Dr. Gillis. I saw a hand raised, so I'm sorry
No
for that.
problem at all. Thank you very much.
Our next question is, you spoke of—
By the way, let me answer Marin with an additional response. My own research in tuberculosis is trying to develop new models of new human models to be used to help predict the response to a therapy and a vaccine that is not an animal. Science is really pushing forward. The director of the NIH is interested in that. My point simply is we don't have that today. And yet we need to cure humans who have ailments. So we have to do this correctly in animal preclinical space. And my answer would be that we need to do this in the proper, humane, and ethical way. So that's the way I would answer that. Thank you, Marin. Go ahead. I'm sorry, Michaela.
Oh, no problem. Great question. Jasmine's asking, you spoke of chronic diseases found in 40% of COVID-19. Could you elaborate on what you have been seeing?
Right. So, a variety of ailments are being seen, and studies and clinics are being developed. You probably have heard about the so-called brain fog. So this idea of mental slowing, lack of clarity in individuals who've been infected over a longer period of time. You're hearing about cardiovascular problems, heart disease, heart failure, some of it related to blood clotting, which has also been associated with COVID-19. itself. You've been maybe very recently hearing about some problems with some unusual brain clots, rare brain clots associated with the Johnson & Johnson vaccine. Chronic fatigue and other organ system dysfunction over a long period of time. In general, I think really we all feel this is linked to the fact that the virus triggers an uncontrolled inflammatory response in some individuals, and that inflammation is underlying some of these more chronic inflammatory disorders, but we don't have all the answers. But I think it's going to be a major problem over a longer period of time once the actual pandemic is more under control.
Our next question is, how much protection can those who are on medications to suppress their immune system benefit from either the Pfizer or Moderna COVID-19 vaccines?
Right. So with the, as I mentioned earlier with cancer, I am saying with a lot of positive response here that in the 30 to 40,000 people in each of the trials, there were immunocompromised, and there was attention on immunocompromised individuals. You know, 30 to 40,000 isn't tens of millions, and that so it has some limitation there. But to date, it appears that the vaccine is working in those with compromised immune systems. And I think this is because it appears for this virus that if one gets enough antibody response against that spike protein, that you can become protected. Now, this is against the wild type strain, not necessarily the Brazilian or South American, the South African strain where the protection is less. So to date, it looks like individuals with suppressed immune systems are still getting very significant benefit from those vaccines.
Our next question is, do you think we need better training in treatment of infectious disease in America?
Yes. Next question. I think we need a lot more education, and I think we need to think creatively about educational programming at all levels. I like to tell an anecdote. At one of my former institutions, the microbiology chair would provide a test to incoming college freshmen. Simple, 1 to 2 sentence questions, and there was always a question on there: define a vaccine. You might be surprised to hear about 10% of freshman students really were close on what a vaccine is as defined. We have a lot of education to do, and I think it's education that is going to help us with my biggest concern, which is vaccine hesitancy here. Let's work together on that. Texas Biomed is an institute primarily doing research, but as someone who's trained many, many students in my laboratory, which is still active today, I focus a lot on the educational components of the institute. We have at least 40 events each year for K through 12. We have undergraduates on our campus. We partner with the medical school and the undergraduate campus on graduate student training. And we do a lot of outreach for the community. This is essential, very, very important. And I always look for partners to spread factual words to the community on infectious diseases.
So I think we have time to take 2 more questions here. The next question is, at what stage in pregnancy can the vaccine be recommended?
Well, the official recommendation is that the benefits outweigh the risks for pregnant women and that they should receive the vaccine. Now, that's not me. That's reading the accreditation organizations within the United States. And so, again, like the immunocompromised host, one could think about pregnancy as being somewhat immunocompromised condition. Vaccine is being recommended. Again, I want to be very careful here and say, please speak to your physician, though, in an individual case about the risk-benefit ratio. It'd be inappropriate of me to be making strong comments to individuals without making that comment. But at the present time, the vaccine is being recommended.
Our last question will be, is there a verifiable method to predict the next cities or locations that could become pandemic clusters?
Yeah, so this is very important. A lesser known story is really a group of predictive modelers in Seattle area who, using global tracking, actually can watch the migration of microbes across the planet in real time and began to see clusters at a very early stage in this pandemic. In developing a program I briefly spoke about that I'm involved with to try to develop a statewide program in Texas, we envision having this global tracking as part of this. I think it's critical, and there's technology out there now to rapidly identify cluster outbreaks that we can then develop a rapid response for. So I think it's a critical question you ask, and one that we need to pay attention to. I think you're going to close us down, and I'd be remiss if I didn't do some shameless promoting of a conference that Texas Biomed is hosting on April 29th and 30th. You can find it on our website. Registration is free. It's called Science, Global Health, and Policy in a Time of Pandemics. We have 50 speakers from around the world. President Bush's daughter will be a keynote, Barbara Bush, who's involved in global outreach efforts, if you read anything about her organization. Tedros from the World Health Organization will be speaking, and we have a number of others from the private and public sector speaking. So please feel free to register and join even part of that symposium. And if we need to close down, I want to again thank you for the opportunity. I'm happy to stay on to hear about the awards if you'll allow me.
Yes, Dr. Schlesinger, thank you again so much for your amazing presentation. There were a few additional questions that we were not able to get to. So if you do have any questions for Dr. Schlesinger, please, you can feel free to reach out to him via email. So we would now like to go into the awards session. With our awards session, we would like to recognize and we want to thank each presenter that was here and shared their work with us today. We can say it definitely was not an easy task for our judges to decide who the poster winners were going to be. But before we announce the winners of our poster session, we would really like to thank the student podium presenters who were selected among the many who submitted for a podium presentation. Each of them will be receiving a $100 e-gift card. So thank you again to our students, Lindsay Roberts, Sharon Sue, and Tyler Jensen. Again, it was so wonderful to hear about all the research that you've done over this past year. For our poster presentation winners, we are going to have the following three categories: research in progress, student presented research, and resident presented research. The first place winners from each of those categories will be receiving a $150 e-gift card. The second place winners from each of those categories will be receiving a $100 e-gift card. And the third place winners will be receiving a $50 e-gift card. So we're going to go ahead and get started with our research in progress category. Our first place winner for research in progress category goes to Melissa Yuko Sperry. So congratulations, Melissa. I wish we could hear all the audience clapping for all of you because I know they all are. Congratulations. Our second place for research in progress is presented to Willem Maniago. Congratulations, Willem. And our third place research in progress is going to go to Courtney Scott. Congratulations, Courtney. We're going to go ahead and move on to our student presented research category. So our first place winner for this category goes to Rachel Ditterich. Congratulations, Rachel. Our second place in student presented research is going to go to Sonia Fan. Congratulations, Sonia. And our third place winner for student presented research goes to Taylor Hutto. Congratulations, Taylor. Moving on to our final category will be resident presented research. For our first place winner in this category, it is going to go to Dushan Harley. Congratulations, Dushan. And our second place winner in this category is going to go to Sai Karan Vamsi Guda. Congratulations. And our third place winner for resident presented research goes to Laurel Shin. Congratulations, Laurel. Congratulations to all of our poster winners and our podium presenters. Each and every one of you did such an amazing job tonight, and we appreciate all of your submissions. I would now like to take the time to pass it over to Dr. Claudia Freitas.
Thank you, Dr. Clark. We really don't have words to say what a feast has been all the symposium, to listen from all the research that you have done, to learn from our keynote speaker and the incredible work that he's been doing, and all those poster presentations. At this time, Dr. Clark and myself would like to take a moment to thank our organizing committee for all their help with making this symposium possible. From building quality service and translating them, thanks Dr. Rollins, to reviewing and accepting abstracts, thanks to our entire committee, each one of you, for creating e-commons webpage and getting a poster uploaded. Scott Park, thank you so much. To creating the symposium website and the program, a big thanks to Jason Roth and Rachel Thomas. Running the Zoom poster sessions, we have our wonderful professors, Dr. Chakraborty, Dr. Mayer, and Dr. Rallis, and their students. Thank you so much. To organizing the judging process, Anna, you've been fantastic. Thanks to you and your students. And also, we want to thank Dan Benson, because she is the one who's going to help us with the award process. We would also like to thank Dr. Talbot and Kendra Angel for working with our sponsors, and Cameron Haidman and his multimedia team for their support through all the planning of the symposium, as well to our amazing judges. Thank you so much. It would not be possible without each one and every one of you. Thank you again. Now, I have the pleasure to introduce and pass the time to Dr. Jeff Talbot, who is the Vice President for Research and Dean for the College of Graduate Studies. Dr. Talbot, the time is yours.
Thank you, Claudia and Micah, for an outstanding event. I'll be very brief as we conclude. I echo, as a university, our appreciation, Dr. Schlesinger, for your time and your expertise, sharing that with us today. A hearty congratulations to all of our participants. I'm going to give a quick shout out, particularly this year, for our first, Dr. Schlesinger, as you talk about education and starting earlier, for the first time at Roseman, we had 2 high school presenters who worked on research, prepared abstracts, submitted through that normal process. Sohini Mandela and Will Maniago, outstanding. So I'm going to share a couple of slides on for Sohini and Will, but really for all of our presenters and those that helped participate. And very briefly, if you ever wonder about the scope of the work that you are doing, I can share this. We started our annual research symposium at Roseman 7 years ago. So in 2014, we started out with presenters just internally from Roseman. I think we had 14 abstracts submitted from each campus, so just 28. And I wanted to again point out to Sohini and Will, but all of our participants, that when we take into account both primary authorship and co-author affiliation, just within the United States, we've had representation from many of our largest and prestigious research institutions in the US. And to pick on a few, the Cleveland Clinic, Ohio State, Dr. Schlesinger, right? We'll work on getting Michigan later. We can have a brawl. The University of Colorado, University of South Carolina, University of Florida, University of Chicago, University of Minnesota, and not to leave out any, but just an outstanding representation of participants in our event today. And it doesn't just limit, it's not limited to just the United States as we look globally. We've had participation from North and South America, Asia and India, Nepal and Malaysia, in the Middle East and Saudi Arabia. Again, just to echo, these are participants in our research symposium this evening. So again, thank you to everyone for your support. I want to take just a final moment to thank our sponsors, doTERRA, a nutraceutical leader on the Wasatch Front in the Utah area, Dr. Russ Osgathorpe and Nicole Stevens, who have generously provided support for our event. Race Swegger and David Kirby from Zions Bank and Practice Pathways, who were introduced earlier. And then finally, a special shout out to Dr. Ryan Young from MedQuest, who is one of our alum. And I think several of these individuals have joined the call. I think Ryan has been on through our event. Thank you for taking your training at Roseman and continuing to serve our communities and now giving back so that we can continue that process. With that shout out, again, thank you, Dr. Schlesinger. Thank you to our sponsors and our participants, and we will adjourn our symposium and look forward to your participation next spring for our 8th annual event. Thank you again, everyone, for your work. We'll see you next year.
Thank you. Bye-bye.